This video is part of a series. It's heavy in scientific terminology and may expect familiarity with previous videos. If you're confused, please watch the rest of the playlist up until this point, or check out my genetics terminology document, both of which are linked in the description. Thanks! So I'm just going to rip the band-aid off now and get it out of the way. Despite, or perhaps because of, the incredible diversity of tails in dogs, we don't actually know all that much about the genetics behind them. It's probably a situation similar to ears, where the sheer complexity of the trait makes it difficult to research, and since dog breeds tend to be pretty set in what kind of tail they have, there's also not a lot of incentive to do that difficult work. But unlike ears, where I found a lot of very scattered, complicated, uncertain info that sent me down a rabbit hole, tails seems like a void of information as it stands. I'll cover what little I was able to find, treat the rest polygenetically, or come up with fictional loci until further info, I suppose. Dogs have a gene similar to Manx and cats called natural bobtail. Dogs with natural bobtail can have any amount of tail, ranging from almost a full tail to a rumpy with basically no tail at all. I don't know the breeder terms, but the sciencey terms are anuri, for basically no tail, brachyury, for a halfish tail, and full tails seem to just be called full or long tails. The variance here is probably just polygenes. I don't see any sources claiming that natural bobtail has the same health problems as Manx, but I'm always skeptical of those simply not being reported. That being said, it's homozygous lethal. It's also dominant, obviously. I see breeders using BT as the allele for bobtail or NBT for natural bobtail, while the recessive long-tail allele is N for normal. However, I see research articles using T for the dominant bobtail instead. Does this stand for tail? Nope, it stands for T-box. The mutation responsible for natural bobtail, C189G, is located in the T-box transcription factor T gene. To explain this a little bit simpler because it's really cool, recall that every single cell in your body contains your entire genome. Your feet know what your eye color is, your liver knows your hair texture, and so on. Transcription factors are proteins in your body, and it's their job to turn genes on or off to make sure the right body part gets read. They're why your lungs don't grow fingers, basically. The T-box is a group of transcription factors that specializes in embryonic development of the heart and limbs, including the tail, which is a limb. The C189G mutation affects the T-box's ability to bind with DNA, affecting tail development. C189G/natural bobtail is not the only gene responsible for short tails. Natural bobtail is the kind of taillessness seen in breeds like Welsh Corgi, Jack Russells, Australian Shepherds, and a bunch of other breeds you'll see in my sources below. Some breeds that tested for not having natural bobtail include, but are not limited to, Boston Terriers, miniature Schnauzers, and Rottweilers. Again, my sources have more breeds for you to peruse. Natural bobtail is distinct from docking. Docking is a surgical procedure done usually to puppies to shorten their tails. Docked tails have an abruptly ending tail with full bone structure that ends in some kind of surgical scar. Natural bobtails have a tapering bone structure followed by a fleshy bit and no scar. By the way, this is not the place to discuss the ethics of docking. I have my own well-researched opinion on docking, and I'm sure you do too. If I see comments discussing docking, even if I agree with them, I will delete them. This is not the time or place to discuss docking. Please keep ethics discussions on topic with genetics, not procedures. Thank you. Speaking of genetics-relevant ethics, the other one I can talk a little bit about is corkscrew tails. I wasn't able to find any genes on them, other than that it is a genetically inheritable condition, but I was able to find some information on them in general that I'd like to share. According to VCA hospitals, the corkscrew phenotype causes a lot of health problems for dogs who have them. The skin folds inside the tight curs may be prone to infection. In severe cases, it can affect the dog's ability to defecate, leading to accumulation of feces in the fold. Not all dogs with curled circly tails are corkscrews in this way, even though the term does get used interchangeably pretty often. Corkscrew, as used by VCA, specifically refers to curls that are far too tight, like these ones in French and English bulldogs. Please correct me if I'm misunderstanding. Another trait that is genetic, even though it's so polygenetic that I can't give you letters for it, is brachycephaly, also known as flat or squished faces, like those found in pugs. You probably know that flat-faced dogs have difficulty breathing and smelling. You may or may not know about all the other health problems brachycephalic dogs face. Problems include, but are not limited to, eye problems, teeth problems, problems thermoregulating, aka they overheat more easily, and so on. The face is an extremely complex structure. You can't just squish part of it and expect everything to continue functioning fine, especially since the rest of the face structures don't scale down with the muzzle. And again, though breathing is a huge one, it's not just breathing. The size of the eye socket of brachycephalic animals is reduced, but not the eyes, leading to eyes that people think are larger, but are actually just bulging out, and in extreme cases, the eyes can fall out. The same number of same-sized teeth are packed into a tighter space, so on and so forth. We're moving on from this now. To another ethics problem, whee! With the cat morphology video, I was able to go back and forth between the ethical and unethical genes pretty consistently, but with dogs, man, everything is just sad. I have two nice ones to end this video on, at least. Let's talk about Shar Peis and why their skin is so fucked up. This wrinkliness is caused by their skin overproducing hyaluronic acid, which gathers under the skin, causing those big wrinkles. Shar Pei wrinkles are pleiotropic with a fever disorder called familial Shar Pei fever, which more or less is what it sounds like. The dog suffers random, sudden fevers, not caused by any outside source. This is because the hyaluronic acid is being misunderstood by the body as a pathogen, so having too much of it is making the dog's immune system panic and start fevering, even when nothing is wrong. Basically, familial Shar Pei is an autoimmune disease. Other problems with Shar Pei wrinkliness largely have to do with itchiness and infections within the folds of the skin. Ugh. The gene responsible for this wrinkly mess has to do with the hyaluronic acid synthase 2 gene, certain to Has2. In this case, the type of mutations responsible are two overlapping duplications. One of them is called traditional, and the other is called Meatmouth. These are named after the two types of Shar Pei, ones who aren't super wrinkly and ones who are, respectively. These genes are very complicated due to the nature of duplications and the way we talk about them, plus the overlapping loci part, so please stand by as I try to oversimplify the important parts. Similar to Merle, covered in a previous video, there's a ton of alleles here due to being potentially lots of duplications of the same set of genes over and over. We're essentially looking at two loci that overlap each other, and thus, one locus mutating a duplication also duplicates part of the other. The traditional locus is about 14.3 thousand base pairs long. You know those A, T, C, G letters? 14.3 thousand of those. Allelic variations of the traditional locus have this set of 14.3 thousand base pairs duplicated a different number of times. The Meatmouth locus partially overlaps it and is about 16.1 thousand base pairs long. Allelic variations of the Meatmouth locus have this set of 16.1 thousand base pairs duplicated however many times. Here's a graph from the article showing the variation here, just comparing different versions of Meatmouth. Control breed refers to non-Shar Pei dogs who have the unaffected or normal allele at this gene. Most of the healthy, non-fever Shar Peis have Meatmouth alleles with fewer duplications. Most of the unhealthy, fever-getting Shar Peis have higher Meatmouth alleles with higher duplications, but there's no strict cutoff point. Put as simply as I know how to break it down, for traditional's alleles, it takes an allele with a high number of duplications before health problems occur. For Meatmouth, it takes less duplications for problems to occur. It's a little easier to picture when you remember that the wrinkles are caused by an excess of hyaluronic acid. The Meatmouth locus codes for hyaluronic acid, so duplicating this region is like the dog's genome accidentally clicking add to cart multiple times per duplication. Complicated, I know, but that's genes, baby, that's the fun part. Sadly, even after all that, I don't have any letters for you, and no dominance model either. I'm starting to notice a trend that the more information I'm able to find about the mechanisms of a dog gene, the less likely I am to find dominance models, though in the case of wrinkly skin, it's probably, at least partially, because the breeds that have them as a standard, so there's not a lot of outcrossing with non-wrinkly dogs to get that information from. Sorry. The way I would simplify this for fictional purposes is as follows. Two loci. Let's call one ST for Shar Pei traditional and the other is M for Meatmouth. The normal, non-Shar Pei versions of each of them are just called N for normal. ST causes thicker skin, but no fevers. M causes Shar Pei wrinkliness and maybe fevers. MM homozygosity definitely causes fevers. And M, even if heterozygous with N, when combined with the ST allele, interact to also definitely cause fevers. Bear in mind that this is a huge simplification and not totally accurate and meant for writers and artists to make fictional characters with. For real scientific info, please default to the source articles linked below. Okay, I said I'd end with two nice ones. Time for some cuteness. Chow chows are known for having blue or blackish tongues. Not just tongues, but their lips and palates are also blue. And they aren't alone, as Chinese Shar Peis have the same thing, and given their evolutionary history, probably from the same gene or genes. The mechanism behind this is easy to explain. Their mouths have excess eumelanin. The genetics, however, are more fuzzy. I've got multiple sources saying it's dominant, though not giving any letters. Make up your own, I guess. But the more reliable ones don't say where the gene is located. One of my sources claims it's an interaction between mutations in two loci, the melanocyte-stimulating hormone gene and the ever-familiar MC1R, which is also where the E and K loci are located. This explanation would make sense as those areas control melanin production. However, this source is just some blog, and they don't cite their sources, so I don't trust them. From poking around other places and seeing how many seem unsure, I'm going to assume that we don't actually know what genes are responsible for chow chow / Chinese Shar Pei mouth blueness. We just know that it's dominant. The same blog also claims that chow chows are born with pink tongues that turn blue around 6 months of age. A more reliable source in the form of a veterinary hospital claims instead that chow chow tongues are blue from birth. A third source claims it develops at 2 to 3 months of age, so I have no idea. It's a common myth that any blueness on a dog's tongue means they have chow chow ancestry. In reality, blue spots and tongues just kind of pop up on dogs sometimes due to all kinds of mutations and all kinds of breeds. Additionally, a chow chow with a pink tongue isn't necessarily a mixed breed. Purebred chow chows are sometimes born with pink or spotty tongues. It disqualifies them from show rings, but they're still chow chows. This one is special to me because my childhood dog had this. The term polydactyly refers to extra toes. The extra toes we'll be covering here today is referred to as preaxial polydactyly, or PPD for short. Essentially, most dogs have 4 toes on their back feet. Some breeds, however, maintain 5 or even 6 toes. Those extra toes are usually called dew claws. I wasn't able to find much on the genetics of any other form of polydactyly in dogs. Sorry. As far as I can tell, dew claws don't cause any problems just by existing, although they can be prone to injury. However, the inductive signaling required to develop digits naturally gets more intense the more digits you need to develop. This causes some negative pleiotropic effects. For example, the gene responsible for PPD in Norwegian Lundhounds may also cause gastritis. And yes, I did word that like there were multiple genes responsible for PPD, in that different ones were found in different dog breeds. Well, that's because there's multiple genes responsible for PPD, and different ones are found in different dog breeds. As for dominance, it seems that the most common PPD-causing gene is dominant. The PPD-causing gene in Border Collies and St. Bernards is recessive, and weirdly, in Australian Shepherds, it's sex-linked and lethal? My childhood dog was a Border Collie mix, by the way. His name was Angel, which is also my girlfriend's name, and no, we won't let her live it down. Being the most common, the one I was able to find the most info on was the dominant one. The mutation responsible is found on chromosome 16 in the LMBR1 region. Essentially, a mutation makes an important signaling molecule called... The molecule... This jump scared me so bad! I was reading scientific articles for the better part of 4 hours, and this just happened to me. I knew about this protein, but I'd never run into it just springing out at me in the wild like a spooky jump scare mansion pop-up. Jesus fuck. Okay, hold on. A mutation makes an important signaling molecule called Sonic Hedgehog express weirdly. There's a zone of the foot during development referred to as the zone of polarizing activity, or ZPA for short, and normally, Sonic Hedgehog expresses there. This mutation makes Sonic Hedgehog also express in the interior limb bud as well, resulting in an extra toe, developing in the form of a dew claw. Yeah, I don't have any practice squares for you at this point, sorry, but here's a bonus for those of you who stuck around to the end. Limb patterned activity and restricted posterior localization of the amino acid terminal product of Sonic Hedgehog cleavage. This is a peer-reviewed scientific paper titled, and about, Sonic Hedgehog cleavage. And on that note, the next video will be a combined extra stuff plus corrections video. The same day and time that goes up, I'll be posting a poll for the next species, and it'll run for 1 week. Until then, bye-bye.